they found there's a decrease in the naive CD4 T cells, so that's expected. And then they had these memory cell populations, and they saw very similar changes in, up or down these memory cell populations in the disease. But what we have in the CD4 T cell population that you don't have in the CD8 T cell population is this TFH, or T follicular helper T cell. And so that typically is located in a lymph node. It's interesting that they find those circulating around, but they tend to be the ones that are helping the B cells make antibodies. So they're indicative, if you have a high level of these, that's indicative of a good antibody response ongoing. And so they did see increased numbers of activated T follicular helper cells in the blood of the COVID-19 patients. But what was, I thought, interesting is they also saw these remaining high in individuals who had recovered. And so does that mean they had some level of ongoing low-level infection, or that there's some sort of mechanism to have that antibody response persist, even though we talked about antibody response is sort of waning, but the T follicular helper cell response seems to be still high. So it's a dichotomy that I can't say I totally fully understand, but I think it is unusual to have an extended persisting T follicular helper cell response measurable following a viral infection.
Yeah, and I think that really highlights a place where there's more research needed in terms of trying to understand the fact that we see this persistent T follicular helper subset and perhaps a decrease in antibody, and how do those things shouldn't go together, but nope. They're happening at the same time. And so what exactly is going on? This might sort of point to something new that should be examined.
Agreed. Yeah. I mean, you can't, these type of papers, you can't argue with the data. They are what they are, right? I mean, they just do this analysis and then just let it happen. That being said, the bioinformatics is not tricky, and two different people doing the analysis could come up with two different things. And so we need to wait and see whether these things are reproducible and whether they look at these in more patients without doing the high dimensionality, but focus in on certain specific subsets without all of the other staining. Does this really hold true for, many, many individuals?
Yeah.
So I think we need to wait on that, but it's still intriguing that there might be something going on. My first thought was, if these T follicular helper cells are circulating, well, then they're not where they need to be doing their job helping the B cells. So maybe there's something about, and they did mention something about some of the chemokine receptors, which they express. Maybe they're downregulating the ones that are necessary to stay in the lymph node. And so then they're circulating when they shouldn't be. And maybe that's somehow affecting the antibody response. But I think there's, like you said, more we need to do to understand that.
I hadn't even thought about that. The idea that the T follicular helpers were lost.
They're just wandering around aimlessly in the body. I don't know, where else my B cell that I need to be talking to, but yeah.
I always look at big data like this as sort of a great way to come up with more questions, more specific questions to then nail down on.
Yeah. The other thing I thought that was really interesting was they started looking at, this Ki-67, so this proliferation marker again, and this time in CD4 T cells and not CD8 T cells. We're done with the CD8s. But so 2/3 of the COVID patients had Ki-67 staining in non-naive T cells. That means they're reacting. So that's good. Their T cells are doing what they're supposed to be doing. They're proliferating. They're then going and helping B cells, hopefully, if they're not lost. But 1/3, that seems like a really high number to me. 1/3 did not have proliferating CD4 or CD8 T cells. So that says that they're really having a defect in their T cell response. Now, one could say, is it lagging? Have they just not quite gotten there yet? So, we don't know the kinetics specifically at this point. But it still is somewhat surprising once you're hospitalized, if you're that sick, that you're not having a T cell response yet. So, Other things that they look at, there was also, the Disney plot, the Disney plots, and they, again, they found certain populations of cells that co-express certain markers, and they did cluster analysis, and they found these clusters, and they could show that according to disease severity, there were increases or decreases in these clusters of cells, or groups of cells, and which ones are driving the disease. I don't know, but the takeaway message that I took was that there was a decrease in this circulating T follicular helper cell population that correlated with severity of disease. So, we talked about there being fewer T follicular helpers, more in the recovered, but there seemed to be a few of these activated ones that were more in the disease patients. So again, is it maybe they're lost, they're activated, maybe they're not quite getting where they need to be yet? I don't know. It was really confusing to me why generally you might have fewer TFH cells, but yet you have an increase in this one that's supposed to be active, and yet you're finding them in the blood where they should be in the lymph node. So
right.
Yeah. I think it's an interesting observation. A lot of this is correlative, descriptive, observational. We really don't know which things might be driving certain parameters, disease parameters, or disease severity at this point.
But as we go forward, and when we get to the sort of specific immunotypes part, then we can sort of come up with some ideas or hypotheses about what we would want to know to figure those things out.
That's right. So the one other thing they did do was they did culture some T cells, which was good. They couldn't do it from all the patients because if you think about this, if they have current SARS infection, that's a BSL-3, and it makes it really tricky to be inducing T cells that, are associated with viral infection. But they were able to culture some of these, and they were able to show that some things were surprisingly not up, like interferon gamma. I didn't see them talking about type 1 interferon, which is interesting because there's a few paper that suggests that type 1 interferon might be defective in these patients. But they did have markers that were consistent with an inhibitory phenotype, like IL-1 receptor antagonist was up, and CCL2 was up, and that's a monocyte attractant, which fits with their increased monocytes that they see in the blood. So, but they were only able to culture the PCR negative patients. So they're severely ill, but they're no longer PCR positive for disease. So we have to keep that in mind as to, how do we interpret these findings that they found.
And one thing that's really sort of interesting there is they saw that sort of low or lower than expected amount of interferon gamma.
Yes.
I wish that they had also looked at TNF.
Yes.
TNF alpha. And the reason why I say that is that there have been a couple of those vaccine papers that Vincent mentioned that do a little bit of the T cell data. And one of the things that I've noticed in some of those papers is that many of the vaccines are inducing T cells that make a lot of TNF and not a lot of interferon gamma. And so it would be interesting to know if it's similar in these patients with more TNF and less interferon gamma.
And so we technically like to see more interferon gamma, less TNF, because TNF is more pathologic, drives more immunopathology. And interferon gamma is better for fighting intracellular microorganisms. So they measured some things and not other things. Some of it might be buried in their data. Maybe they chose not to, show it. I didn't look and see whether TNF was in their panel, because if it is, I assume they've deposited all of their data. And if one were, bioinformatically savvy, you could go and interrogate those data and find out a lot more information about your markers of interest.
That is true.
Yeah.
I hope one of my students isn't listening right now, so he doesn't know that something we can talk about.
Wait, so that you can bring it up and seem smart, or?
Yes, exactly.
See, one of the trainees, you don't understand that the PIs are always trying to stay one step ahead of you. So that they're like, oh, did you see this paper? And they're like, no. Oh, yes, I found one I couldn't find.
Hey, we get listeners sending us papers all the time.
Yeah. Secretly, though, we're hoping that you beat us to the punch and that you find the papers and send them to us, because then we know you're doing really, really well. So they found lots of cool things with CD8 T cells and CD4 T cells. But of course, people want to know what's going on with B cells, because most of the vaccines we're going to make are related to B cells. So you'd like to know, are B cells, are the antibodies that we're making really important? Are the B cells defective? How good is this going to work? So, they did, photocopy again, next page. It looks almost exactly the same. And if you didn't know your markers, you might think that they're all on the same thing, but they start looking at B cells, and they of course look at multiple different B cell populations. And this is one place where we could probably take a moment to talk about this. And so, again, there are naive B cells, just like there are naive T cells. They have not seen antigen. They're not yet making a lot of specific antibody, but B cells are different than T cells because they can make some antibody even before they're stimulated, right? This is the natural antibody. And they will express IgD and IgM, but you can use IgD and IgM markers and what they're expressing to sort of look at whether they've been activated or not. So once they're activated, they usually downregulate IgD and upregulate, other antibody isotypes, and they also class switch. So you can look at, where are your antibodies along the spectrum of activation? If they're making if they're surface IgD positive, they're usually naive or very early after stimulation. Then they lose IgD, but they can still make IgM, and they'll make IgM at the beginning. And there's some B cells will make tons of IgM until you get some class switching going on, which means then you start to make IgG. And usually we think of the IgG as the workhorse, right? That's the one that really is good at neutralizing. And also during this whole process, which we could go into a little bit more detail if you want. But remember, these B cells are really cool in that they can also muck with the sequence of the antibodies that they make to try and make them better. So they fine tune the sequences so they can make stronger affinity antibodies. And so they're at the same time, they're class switching to make antibodies with different functions. They're also mucking with the sequence to make higher affinity. And they have to go through selection in the germinal center to make sure they're not autoreactive and so forth. But you get these class switched antibody expressing B cells. You also get memory B cells. And then we also talk about a class of B cells called plasma cells. So they basically are just antibody factories. So they've gotten their right class switch, they've gotten their right sequence, and they got the go-ahead signal, and they just turn into a factory and just make tons of rough ER, and they just churn out huge amounts of antibody per cell on a per day basis. And so those are the plasma cells. And then the precursors to that are the plasma blasts, which are proliferating a lot because they've gotten that signal, and then they're going to start to churn out the antibodies.
Yeah, and so this is sort of paralleling what we saw with the T cells, in that we're able to make different subsets of the B cells that are at different stages sort of after activation, and they're having slightly different functions. With B cells, we've done, at least with the B cell subsets that are listed here, there is a little bit more of a tie to function.
Yes.
With some of these different subsets, as Cindy sort of just nicely talked through. But it's the same kind of concept that we're looking at within the B cells, at cells that are at different stages of activation and sort of different stages of their life.
Right. And so there were differences, up, downs, whatever. But I think the key takeaway was there was just a massive expansion of these plasma blasts, which are the ones that have sort of gotten all their signals and they're proliferating, and then they're going to turn into these antibody factories. And so it 2/3 of them had this increased plasma blast, that, these hospitalized patients, but that means 1/3 of them didn't. Right? Which is also interesting, because that might account for why there might be a proportion of the patients that are not making antibody responses. But this isn't that surprising, because it's not unique to SARS-2. This is something well described for other viral infections like dengue and Ebola and so forth. You have an acute viral infection, you get a strong antibody response, you're going to have a lot of these plasmablasts circulating in the blood. So that wasn't all that surprising. They definitely saw expected features like increased proliferation of all the B cell populations. They saw clonality, which I thought was really cool because that suggested that there's antigen-specific responses happening.
So what that means is that the clonality means that there was sort of one initial B cell that was triggered when it saw antigen, and it divided and divided and divided and made lots of progeny. And so all of those B cells have the same genetic sequence. It's not as if we have that sort of random bystander activation where excess cytokines or excess other sort of immune factors are just triggering any old B cell. The clonality really tells us that these are the B cells that are specific to SARS-CoV-2.
Right, which is good. You don't want random B cell populations expanding out. You really want to put the energy into the antigen-specific ones developing. So that's good. One of the other key features they saw was a reduction in a chemokine receptor that's required for homing cells to the lymph node and B cell areas. So that's a little interesting, right? Because we want the B cells to be in the lymph node and getting the T cell help and making the antibodies and doing their class switching and affinity maturation there. But they have lost that marker, that homing marker, and now they're circulating around the blood. So are they lost again? I don't know. There seems to be something happening.
And that makes it tricky because to make high quality antibodies, those B cells need to interact with CD4 T cells. And if the B cells are lost and the T cells are lost, they're probably not going to be able to interact to make the best possible antibody response.
Right. The other weird thing was, you brought up the T cells, is we talked about those T follicular helper cells being really important for driving the B cell responses. One would think that if you have a whole lot of plasmablasts, you should have a really good T follicular helper response. But remember, we said that was actually down, although there was a subpopulation that was activated. So it's interesting that one would have anticipated if you have a high plasmablast, you should have high T follicular helper. And it doesn't seem to be the case. So there's some disconnect between the components of the immune response that should be working together based on everything else we know. And they don't seem to be working as we expect them to be working in this disease, whether that's driven by the infection or whether we have a magnifying glass that we can now see things that we couldn't see before, and those differences are popping out that we didn't know about before. So it's interesting. So the weird thing to me, and I'm not sure, maybe I missed something as I was reading through this, but they seemed to say there was not a good correlation between the good functional antibodies and the plasmablasts. So it seemed to me that they were saying that they got lots of these plasmablasts, but they don't seem to be making the right antibodies that they need for this infection.
Yeah. So they were specifically talking about antibodies that were binding spike there.
Right.
And so they kind of indicate, well, maybe we're getting lots of antibodies to something other than spike, or maybe, there's some other things going on. Maybe, we're looking at other classes of antibodies or something like that. So they hand wave a little bit at that.
Yep.
And that caught my eye because I had been paying attention to some other literature that's been talking about spike versus other antibodies. And so the idea that perhaps they were missing some of the antibodies, maybe the plasmablasts were really correlated with the nucleoprotein antibodies or something like that, could be sort of interesting to me, at least.
Right. Yeah. I don't know. So I don't want to go into too much more details about this paper, but start to back out to the kind of the bigger picture. And that was, so they did something like, it was the first time I'd ever heard this term, the Earth Movers Distance, or the EMD. I thought, wow, that's really cool. And I can't say that I fully understand how they did it, but they just took this like hugely multi-dimensional data and just, asked it to group clonotypes or immunotypes of COVID patients. So could we actually say that some are more severe, and do those more severe ones have a specific immunotype? Can we use that to predict how they're going to, how the disease is going to progress?
Yeah. And this is sort of related to the clusters that we saw in the T-SNE plots before. But basically what they wanted to see was whether patients who had different severity of disease had enrichment or reduction in some of those fractions they made in the T-SNE plots before.
Right. And in this way, they're not just looking at one parameter at a time. So it's not just like, okay, this patient had more severe disease. Let's go back and look at this particular T cell population or this particular B cell population. But it sort of looks at it at a whole, and it asks like, what are the immune drivers that correlate with the severity of disease? So it's not just one aspect. So do they have a failure in B cell proliferation plus more of certain types of T cells, or exhausted T cells? So is there an immunotype? So is there a set of immune parameters that define a particular population of people with disease? So one of the things that they were able to do when they were doing this analysis was they took a subset of their patients and were able to take their day 0 data, but then sample them again on day 7 and see whether these parameters were stable or whether they changed. And might that indicate whether these immune parameters kind of drove those responses? And so generally what they said was that pretty much everything was stable to some degree, but there were some things like that might be expected. So the frequency of non-naive T cells went down over time. So we would expect that because you'd have more responders. And then they did things that I'm not sure I fully understand, but they used healthy donor data to determine variabilities in parameters, because one of the notorious things with human studies is there's just a huge variability. And so they used healthy donors to define variability, and then used that to actually analyze their data to get rid of the noise and see what parameters were driving these different immunotypes. And so they found that of the diseased patients, like 50% of them had an increase in the number of non-naive CD4 T cells. So that makes sense. And 30% had no change, and 20% it actually went down. So they could group these into, like, for example, just looking at CD4 T cells, 3 immunotypes, right? So some responded better. So that means they were on an upward trajectory. They were not responding well at first. They had a better T cell response later. Some were the same, and some actually went down. And so the same was true with proliferating CD8 T cells. Some of them had no change in response, and some of them went up, and some of them went down. And so there's lots of ups and downs and differences, but the question of what does it all mean in the end? And so they were able to do one more dimension of analysis. And that was they took each one of those up downs and their immunotypes, and then they correlated that with all the different parameters that they had looked at. And they asked, is there anything that seems to change between day 0 and day 7 that is associating with driving disease? And so what they saw was that CD4 T cells going down correlated with an increase in white blood cells and neutrophils, that decreases in T cells correlated with increases in Apache scores. So what does this all mean? So this just means that if you lose your T cells, you're going to get sicker. Which now we come back to saying, how does that fit into what everybody's saying? There's some camps that are saying the disease is immune-mediated pathology and more is worse. And this is actually saying losing your T cells is worse. So it may be in this case that you need the T cells to control the disease, but yet expansion of other populations are actually driving disease. So, what the intricate little things that are coming out here, I don't know if the details are important, but just being able to take the information that we're getting and look for trends that go up or go down or correlations are important.
Maybe as you lose T cells, then you have too much virus, and then that gives you more cytokine-chemokine responses, and it's all funneling.
More immunopathology, right?
Yeah. And so they make these three groups, these three immunotypes. Group 1 that has a lot of CD4 T cell activation and proliferation,
right?
Activated and exhausted CD8 cells, and a certain type of plasmablast. And those individuals seem to have increased severity. There's a group that has group 2 that has sort of less of a CD4 response, more activated and replicating B cells. And then there's group 3 who as, and I quote, an immunotype largely lacking detectable lymphocyte response, suggesting a failure of immune activation.
So which one do you want to be in? Which immunotype? 2?
In some ways, 2, but 3 seems to have pretty good outcome after disease.
Right.
And so what you'd love to be able to do is to determine whether there are differences between these 3 immunotypes and response to treatment. Is there some treatment that's better for somebody who's in type 1 versus type 3?
Right.
Or if you could somehow take this, and this makes my brain hurt a little bit because I'm imagining just how much big data analysis would have to go into this, but take this and then look at, say, genetic associations. Are there genetics that make you more likely to be in group 1? Can we can we say, oh, if you have this genetic polymorphism, you're going to be in group 1. We need to sort of take care of you earlier, or something like that. Being able to overlay that onto this idea of immunotypes could be really useful, because it could help us predict disease, or knowing this could help us understand better treatment options.
Oh, that sounds like it's a ways off.
Yeah.
Yes and no. I mean, they were able to at least, some of these patients had different treatments like hydrochloroquine or remdesivir. And so you could look at, do those treatments correlate with any of the immunotypes or any of the parameters that we know would be beneficial? And they didn't seem to see anything.
Right. Yeah. So
in their data set, it didn't seem to make any difference one way or the other.
So maybe if we had some other treatments that, seem to be working a little bit better, perhaps they would be more clear here.
Right. And coming back to which immunotype you want, I mean, according to their data, like Brianne said, I mean, the third group is, it seems like their response sucks, but they don't have the immunopathology, and their disease is less severe. So for whatever reason, maybe they're early controllers, and so that they don't have as robust a long-term response. Then, the big question comes, do we want a vaccine that induces a robust response? Because that's the big question that everybody wants to know. So, because those that had a more robust response actually did worse.
If you mentioned that some individuals who regulate virus early, so what's regulating it? It's not a B and T cell response then, is it?
It might be more, maybe their innate immune system is working. Let's go innate immunity. Woohoo! We both really like the innate immune system. Maybe the innate immune system produces a whole boatload of type 1 interferons and innate immune responses and controls things really early before you ever have a chance to have a robust adaptive immune response.
Yeah, because the timing is consistent, because Michael Minnis said most patients who recover and don't get serious disease, the virus is down by day 7 post-symptom onset, and they don't transmit it anymore. Can't be adaptive, right? Or mostly not adaptive.
It could. I mean, by 7 days. Yeah. I mean, so everybody sort of says 7 to 14 days for antibodies, but really, I mean, that's a little long.
That's a little late.
But
3 to 5 days is when you're going to start. I always, when I teach this, I'm like, if you've ever had the flu, you're going to start to feel a little better 3 to 5 days later, right? It's peaks and then you start to feel better. And it might, go on for a couple of weeks if flu is really bad. And I imagined it originally that this was going to be like that. But, so by 3 to 5 days, you're starting to generate B and T cells. So maybe it's just that initial response was enough to control it. So without the continuing antigen, you don't have continued tickling to make sure you get a robust response. And it just wanes.
I think that the looking at these 3 immunotypes brings me back to sort of the one caveat from the beginning of this paper, which was that most of the patients they looked at were patients with severe disease. They don't really have a huge number of, say, asymptomatic infections or things like that. And so do those patients fall into one of these three immunotypes, or do they have their own immunotype that is actually the one you want?
Yeah, that's really important, right? And that's because they're just capturing patients coming to the hospital, right?
Right.
As opposed to setting up a trial, which would be hard, right? Screen people. You're going to screen people and pick out the asymptomatic. Wow, it's really tough. You're going to get them all different.